Welcome to the deep dive. Today, we are taking, well, one of the most fundamental biological processes, seeing and looking at it through the lens of physics, chemistry, and biology. Basically how vision works, not as magic, but as this fascinating chain reaction.
Absolutely. And the source material we're using today is just fantastic because it tackles sight with this real sense of wonder, but focuses hard on the basic principles. Our goal here is really to follow that signal.
Right.
From the moment a photon, just one photon, hits the eye through all the optics, the chemistry, right up to the neural processing that happens even before the signal hits the main part of the brain.
It sounds like sight is much more about interpretation than just, recording an image.
That's exactly it. Interpretation versus simple reception.
All right, so let's start with maybe the most surprising part.
Yeah.
Color. I think most of us assume, red is red because the light is red.
Right.
But it sounds like it's messier.
Oh, much messier. And what's really striking is how fast the brain starts, well, adjusting things. Take the blue shadow example. It's a great thought experiment.
Okay, walk me through it.
So imagine a screen. You light it up with two light sources, one pure white, one blue. Then you put something in front to cast a shadow, but arrange it so only the white light hits the shadow area.
Okay, so the shadow is getting only white light. It should look white, right?
Logically, yes. But to our eyes, it looks blue.
Why?
Because the whole scene is bathed in that blue light. Our brain basically says, okay, blue is the default here, and it sort of subtracts the blue from everything. It sees the white light on the shadow as lacking the background blue, so it interprets it as, well, bluish. It shows color is relational, context matters.
That's wild. And then there's Land's experiment, which really threw a wrench in the works for the simple color mixing ideas.
Oh, completely. Land basically proved you don't need all the colors of light to see all the colors. He took two black and white photos, right? One shot through a red filter, one through a green. Then he projected them on top of each other. One projected with red light, the other with, say, white light, or even just green light. And people reported seeing a full range of colors, blues, yellows, everything, just from those two initial images and specific lighting.
So the standard theory, the trichromatic one, where we have three types of comb cells in the eye, one for red, one for green, one for blue, and the brain sums up their signals. That's not the whole story.
It's the starting point. That's definitely how reception begins. You do have those three cone types, roughly sensitive to those parts of the spectrum. But what happens next, as soon as that signal leaves the cone, is where it gets really clever.
Okay, what happens next?
It moves into what's called the opponent process. Think of it as like a data compression step happening right away in the nerves.
Yeah.
Instead of sending three separate signals, this much red, this much green, this much blue, the nerves carry combined antagonistic signals.
Antagonistic, meaning opposite.
Exactly. You have nerve fibers that signal red versus green. If red light excites it, green light inhibits it. And other fibers signal yellow versus blue. It's incredibly efficient. It means the eye is already analyzing and compressing the data before it even leaves the retina.
And that's why we can't perceive like a reddish green, because those signals cancel each other out in that pathway.
Spot on, or a yellowish blue. The wiring itself prevents those combinations. It's all about contrast and efficiency.
Okay, so the interpretation starts immediately. But let's talk about the hardware, the eye itself. It's often called backward wired, right?
It really is one of biology's oddest designs, structurally speaking. The retina, the light-sensitive part, is technically an outgrowth of the brain.
Yeah.
But yeah, the light comes in, passes through the pupil, and then has to travel through several layers of nerve cells, ganglion cells, bipolar cells,
Before it gets to the detectors.
before it finally hits the rods and cones, which are way at the back of the retina. So the wiring is literally pointing away from the incoming light.
Doesn't that, mess up the image? Passing through all those cells first?
You'd think so, but apparently those nerve cells are incredibly transparent. The loss of quality is minimal. The blind spot, though, that's related. That's the spot where all those nerve fibers bundle together to exit the eye and go to the brain. There are no photoreceptors there at all.
Got it. Okay, so focusing. I always thought the lens did all the work, but the source material emphasizes the cornea.
Yeah, the cornea is the real heavy lifter for focusing. It's that outer clear layer because it's going from air, which has a low refractive index, to the cornea, which is mostly water and has a much higher index, like 1.37.
That big change in density bends the light a lot.
Exactly. It does maybe 70% of the total bending right there at the surface. The lens behind it, that's more for the fine-tuning. The ciliary muscle changes the lens shape just enough to adjust focus for different distances. That's accommodation.
And the iris, the colored part, that's like the camera's aperture, controlling how much light gets in.
Right. And adjusting depth of field, too. But let's get down to the rods. Night vision. This is where physics meets chemistry in a really direct way.
Okay, tell me about rhodopsin.
Rhodopsin, or visual purple. It's the pigment in the rod cells, made of two parts, retinine and opsin. And the sensitivity here is just staggering. A single rod cell can fire off a nerve impulse after absorbing just one photon.
One single quantum of light. How?
It's down to the shape of the retinine molecule. It has a specific double bond. When a photon hits it, the energy is just enough to break the constraint on that bond, causing the molecule to physically snap into a different shape. It's called a trans effect.
So a tiny change in molecular geometry triggers the whole nerve signal.
Precisely. That shape change sets off a chemical cascade, amplifying the signal and leading to the electrical impulse sent to the brain. It's an amazing conversion of light energy to information.
And that sensitivity, is that why bright light blinds you for a second when you come out of the dark? You've built up too much rhodopsin.
That's exactly it. In the dark, your rods build up a big supply of ready-to-go rhodopsin. You become dark adapted. Then, boom, bright light hits, and it bleaches out that rhodopsin faster than your eye can remake it. The system is just overwhelmed for a moment.
Okay, so the signal's generated. Now it has to get to the brain in a useful way. Both eyes see slightly different things. How does that get combined? The optic chiasma.
Right, the optic chiasma. It's this crucial crossover point behind the eyes. Here's the clever part. Information from the left side of your visual field, which includes signals from the inner half of your left eye and the outer half of your right eye, all gets routed to the right side of your brain.
And the opposite for the right visual field, going to the left brain.
Correct. This ensures that everything you see in one spatial area gets processed together in the same brain hemisphere, creating a seamless picture.
And this wiring is incredibly specific, isn't it? The salamander example from the lectures is just mind-blowing.
It really is. It asks, how fixed is this wiring? So experimenters took salamanders, cut the optic nerve, rotated the eyeball 180 degrees, and then let the nerve regrow.
Okay. So did the nerves rewire to match the new orientation?
Nope. The nerve fibers grew back and reconnected to the exact same spots in the brain they were originally wired to. They didn't care that the eye was upside down.
Meaning the poor salamander saw the world permanently upside down and backward.
Exactly. It's a powerful, kind of sad demonstration of how predetermined some of this neural mapping is. It prioritizes the original map over functional adaptation, at least in this case.
Wow. And even controlling our eye movements is split. We can choose to look at something close, making our eyes converge.
Right, turning them inward. We have conscious control over those nerves.
But scanning side to side, moving both eyes together.
That's a different set of nerves, a different system. We don't have the same direct, voluntary control over that parallel movement. It's more automatic.
Fascinating. Okay, let's step outside the human eye. Insect vision, like bees. Totally different setup, right? Compound eyes.
Completely different. Instead of one lens, they have hundreds or thousands of tiny individual units called ommatidia. Each one is like a mini eye with its own lens and detector.
It gives them a huge field of view, I bet.
Massive field of view. But the trade-off is terrible resolution, visual acuity. Each ommatidium is tiny, maybe 30 microns across, so the detail they can resolve is very limited compared to us.
But they gain other things, like seeing ultraviolet light.
Yes. Bees see down to about 2,900 angstroms, well into the UV, so flowers look totally different to them. Many have UV patterns, like nectar guides, that are invisible to us.
And they use polarized light for navigation.
Right. They can detect the polarization pattern of sunlight scattered in the sky, which helps them navigate even when the sun isn't directly visible. It just shows evolution uses whatever physical cues are available.
And just quickly, the octopus eye, it's like ours, but wired the right way.
Sort of, yeah. It's a camera-type eye, evolved completely independently, similar structure lens, iris, retina, but the light hits their photoreceptors first, without having to pass through nerve layers. It's wired right side out, you could say.
Okay, finally, let's talk about the frog. The ultimate example of the eye not just seeing, but analyzing.
This is maybe the most stunning part. The frog's retina does an incredible amount of processing before the signal even gets close to the brain. It's filtering and interpreting like crazy.
They found specific types of nerve fibers coming out of the frog's eye,
Right.
each doing a different job.
Exactly. 5 distinct types were identified, which tells you the retina is already specialized. First, there are fibers for sustained edge detection, just seeing outlines and boundaries.
Okay.
Second, the famous convex edge detectors. These fire most strongly for small, dark, curved shapes that are moving, basically bug
Makes
detectors.
sense for a frog.
Totally. Third, fibers for changing contrast. They respond to things moving or flickering. Fourth, dimming detectors. These respond when the overall light level is decreasing.
Like a shadow passing overhead. A potential predator.
Could be. And fifth, darkness detectors. These just signal when it's dark a sustained response.
So the frog's brain isn't getting a raw picture. It's getting alerts like bug moving at position X or shadow overhead.
Pretty much. The retina is doing heavy-duty filtering, pulling out only the information deemed most relevant for survival and sending these highly processed reports upstairs. The compression is immense.
That really changes how you think about seeing. Okay, let's recap. We covered color perception, how it's relational and processed through opponent channels. We looked at the eye's optics, the cornea's power, the lens's fine-tuning, and that weird backward wiring.
We dove into the chemistry of rhodopsin, how one photon triggers a signal. We saw the fixed wiring with the salamander example, and contrasted the human eye with compound eyes and the octopus eye.
And finished with the frog's retina as this incredible preprocessor, filtering reality before the brain even sees it.
Yeah, vision as this cascade, physics, chemistry, optics, and intense neural computation, all starting right in the eye.
So the final thought for you, our listener. If seeing isn't just passively receiving light, but this incredibly edited, compressed, pre-interpreted narrative, if the signal is filtered so much based on evolutionary needs before it even reaches your conscious awareness, what does that really mean about the objective reality you perceive every day?
Definitely something to think about next time you look around.
Thanks for joining us for this deep dive into the physics and biology of sight.